So with that, let me introduce Dr. Joe Connors. So Joe Connors is a medical oncologist at our BC Cancer Agency, and he's director of the Center for Lymphoid Cancer. We have a very close working relationship over many years with Dr. Connors, and are working on a project right now, a very significant project for Canada, and for that matter, the rest of the world in lymphoid cancer. So with that, let me introduce Joe and turn it over to him. So, thank you.
Thanks very much, Brad. It's a delight to have a chance to speak with all of you this evening. I get to talk about stuff that I find fascinating and wakes me up in the morning and sends me off to work eager. And so I'm going to try to first characterize why cancer is such an important problem in BC, Canada, and the world, and then a little bit about how medicine and scientists think about cancer today, and tie it into the kinds of research techniques and investigations that Brad was describing for you. So I wanted to start off with the kind of the big picture, because cancer is a genuinely worldwide problem. These shaded areas talk about the incidence of cancer around the world, but what I want you to notice is that no continent is saved from colors, and this tells you that it's a worldwide problem. Every population, every ethnic group, every separate society is experiencing the dilemma of dealing with cancer as a major medical problem. The worldwide statistics are in many ways daunting. One reasonable estimate is that between now and 2030, we're going to go from about 14 million cancer cases per year to 24 million worldwide. And that's going to lead to around, in this year alone, about 8 million people. There are some 30+ million people living with cancer, the consequences of it and the treatments they've received for it in the world today. And it's the leading cause of death in the developing world. It's, in the developed world, it's quickly becoming the leading cause of death in the developing world. So we share this with everybody else on the earth. In the developing countries, about 1 in 2 people in their lifetime will develop some form of cancer and need to have that dealt with. So this is a common problem that's going to affect half of all of you here in this room. It's going to affect me and everybody else in our country. And I'm going to come back to the consequences of cancer as it plays out into becoming a serious disease, because you can see that the lifetime probability of dying from cancer in Canada now is getting up close to 1 out of 3. So there's no escaping it. This is a problem that we're all going to have to deal with. Huge amounts of progress have been made across the disease spectrum of various kinds of diseases over the last 50 to 60 years. And to get some sense of just how challenging the problem of dealing with cancer is, it's worthwhile looking at how much progress has been made. And so you can see the decrease in the mortality rate from heart disease, cerebrovascular disease, those are strokes, infectious disease and pneumonias, and respiratory infections. And you can see these dramatic drops that have taken place. And the challenge with cancer is just how, is shown up very nicely there by how difficult it's been to actually bring that mortality down. And so part of what we're embarked on using genomics as an investigatory tool is to see if we can't try to catch up with those other areas of medicine. Another very important thing to understand about cancer is its relationship with age. I've taken as an example the area in which I work, non-Hodgkin lymphoma. And these are the ages across here and the incidence, the number of new cases of cancer against those age. And so you can see that from the pediatric area here, that with children, we rise up here to the 60s and 70s and 80s, an enormous increase, about a 50-fold increase in the incidence each year by age group. And so that tells us, it's a very important clue, because it tells us this cancer's got something to do with the passage of time, with the aging of the cells in our body. The impact on society plays out in a number of ways. It's contributed to by 3 different things. There's the rate change. Some cancers are growing more frequent, but the population is getting bigger. And the age of the population is going up, and I just told you that older people are the ones that get cancer. And those accumulate to that top line, and so it's reasonable to anticipate that Canada and countries like ours are going to have to figure out how to deal with twice as many cancer patients every 15 to 20 years. I've been here for 30 years, and so I've seen a fourfold increase in the number of cancer patients that we're looking after here in BC. So to think about this problem, I'm going to go through some basic biology. So we have to remember that our bodies are composed of many different kinds of cells. All of these cells have a specific purpose. This is just an anatomic diagram cut away to show the different kinds of cells. We've got lymph nodes and teeth and muscles and bones and nerves and blood vessels. All these cells have to cooperate. They have to get along with each other. They have to pull together. They have to keep us alive and making everything work properly. So if I jump down to the cellular level, I cut right down now to a biopsy from a piece of tissue, and these individual cells are talking to each other constantly, communicating with each other, and they're following a set of directions that are intrinsically programmed into them that tell them what to do, how to play out their lives. So in each of us, each adult human being has about 70 trillion cells in their body. That's a lot of cells to talk to each other. Every day, somewhere between 500 billion and 1 trillion of those cells die off and have to be replaced. So that means we have to grow new cells very vigorously every single day of our life in order to stay the same size. Most of you are probably about the same size as you were 20 years ago, maybe a little bit bigger, but about the same size as you were 20 years ago. So you've been replacing these cells every day at the rate of around 1 trillion new ones. You can't do that in a haphazard way, and so we have to get rid of the old cells in a programmed way, and that's what cells have built into them, a process called apoptosis, or programmed cell death. Every day, a bunch of cells on purpose die off, and a bunch of other cells come into being. Somebody has to be running the show. Something has to be coordinating all of that activity. So a way to picture just how much information we're talking about here is, if you don't do it because you'll poke someone in the eye, but if you imagine if you spread out your arms like this, the distance between your fingertips is a meter or two. Well, if we stretched out a molecule of DNA, the molecule that makes up our genetic information, it would be about that long. Now imagine that that was a long string with beads along it to program the instructions, and you started twirling it and twisting it till you can kind of compress it smaller and smaller and smaller, until you finally manage to twirl it and squeeze it down in size so that it would fit inside one cell. Now imagine that you have a freckle on your body. There are about 1 million cells in a freckle. So we've got to pack that 3 feet, that meter of information, of string that's all packed together into 1 million of those in that little tiny space of a freckle. To do that, it's twisted and turned and wound up into structures like this so that it can be packed into something that size. Yet, those are the instructions that run the cell. The information packed in there has to be gotten at and paid attention to every day. So now I'm going to drill down to the size of to the string itself and look at one gene, one string of instructions along the gene. They have a basic structure. If we talk about that whole gene, there's the actual information, what to do, the instructions, and then there's a promoter that turns it on, that says, pay attention to these instructions right now, and regulators that turn it off. So you have on-off switches to tell the gene to be, the information to be paid attention to and something done. These genes are on this, that's the string that I just told you we're going to wind up tightly into tiny little spaces. And those twisted together structures are called chromosomes. We each have 23 pairs of these chromosomes. And ordinarily, we could take a picture of them using special microscopic techniques and look at these 23 pairs. And the ones taken from this patient have a couple of problems with them. You can see there's an intact chromosome. There's a chromosome with a big piece missing from the end of it. Here's a problem where a whole extra chromosome has been made, a trisomy, a triplication of that particular chromosome. So we're going to come back to why that has consequences by drilling down to pictures like this, where we, instead of just black and white, we can color the chromosomes with specific colors, and then we start to see that this green chromosome has a bit of blue chromosome stuck on the end. So in taking the chromosomes apart and putting them back together again, this person's cell has made a mistake and tacked on a piece of chromosome in the wrong place on the end of a chromosome. Well, what consequence might that have? So let's blow up one of those chromosomes. This is a very abnormal one here that's been assembled out of the pieces of chromosome 9, 1, 9, and chromosome 3. This is an improperly assembled structure that's made of pieces of chromosomes. Now I'm going to drill down to the reason why that's important. So I told you that every day we have to get rid of many, many cells. We choose which ones to throw away because they've become genetically damaged. They've aged, things have broken down. We have to get rid of them. And when those cumulative genetic errors build up, we send a death signal into the little energy packets in our cells called mitochondria, and they start leaking. And these chemicals called cytochrome C leak out of the mitochondria, and the cell reacts to that and says, it's time to give up the ghost, I'm going to commit suicide, and that cell undergoes cell death. Well, if I go back to one of those chromosomes here, where I've got a pair where a piece is broken off the end of this chromosome and gotten tacked on to the end of a different chromosome, we've got this translocation. Piece here has gone here. Well, the problem with that is it moved a gene, which was sitting on the end of that chromosome, to the wrong place. And in this particular case, it was the gene called BCL2, for B-cell lymphoma gene number 2. It's in the wrong place, but it's intact. It's just being turned on because it's now next to that promoter that I told you was the on-off switch, and it's being left on all the time instead of being turned on and off the way it should be. And the consequence of that is that too much of this BCL2 plugs up the mitochondria. It can't release the cytochrome C, and so the cell doesn't know to undergo its programmed cell death and doesn't disappear. Well, this is an old, broken down, obsolete cell with a lot of genetic errors in it. And having it stick around is a bad idea because other things can go wrong with it. And so I'm going to close my description of all this by talking about how the body tries to deal with that kind of problem. So all of us have these cells called macrophages here, this big gray thing. It's creeping around on top of a glass slide in this picture. And as it did so, it encountered a germ, and it ate the germ, and the germ is inside being digested. And this other cell is sitting on the surface of that macrophage, listening and watching what happens when that germ gets chewed up, memorizing it, and taking that information off to the immune system and providing us with immunity against that particular germ. That's the normal way we resist invading germs in our environment. Those same cells that learned in that case from the macrophage how to attack the germ can also be used to attack cancer cells. And so how you can see a swarm of these little blue cells here. These are what we call cytotoxic T cells, or lymphocytes, or effector cells in our body. And they don't like this big cancer cell here. They recognize there's something wrong with it, and they've swarmed all over the surface of it, and they're poking holes in it, trying to get rid of it. So this is what we think at least protects us from developing some cancers, because these cytotoxic T cells have realized that cell made a bunch of genetic errors, hasn't been destroyed the way it was supposed to be, and needs to be gotten rid of. So we go searching for the regulators of this. And now I need to have you picture that long string again, and I want to figure out what the signals are all along the string. And scientists can do that by what we call tiling. We take a little short section of that genome and duplicate it so we can examine it, and we keep moving the section that we look at along the genome until we come to a place where there's a skip. Because this section has been tiled, it's reaching across a place where it should have been all chromosomes 6 or 9 or whatever, but there's a jump, and it's jumped over to a different chromosome. So these two genes here have been put next to each other when they shouldn't be next to each other. That consequence is that it has an effect on the immune system. So here's the cell, a malignant cell that's got that translocation, the genetic material's been moved to someplace new. We've tiled along it and we've found out what's new about the structure of that gene. And it turns out that there's a gene called C2TA that's been moved to the wrong place. It's now being transcribed or expressed in that wrong place. And when it's turned on, it sends out a signal and it turns off the signal that allows that cell to be recognized. And that cell that just disappeared off there to the right was supposed to attack that cancer cell and can't do it any longer because that gene is not expressed on the surface. A different gene has been translocated in the same cell, and this one's called Program Death Ligand 2, PDL2. And when that gene is made by this cell, it expresses a molecule that goes over here and tells the destructive T cell that's supposed to get rid of that cell not to do so, and so he's taken out of the picture too. And now the cancer cell is hidden, can't be seen, and disguised so that the effector cells can't find it and do something about it, and manages to linger on in the body. And both of those events were programmed by that cytogenetic abnormality, that redistribution of the genes within the cell. So today, we're tackling that problem straightforwardly because we have figured out that this malignant cell ought to be attacked by this effector T cell, but it's being blocked from doing so by the production of that Program Death Ligand 2 blocker that's keeping this from happening. And we can take another antibody and stick it in there and absorb away that disguising chemical. And so figuring this out took the need to understand the basic genetics of the cell and then start to manipulating it by using its own machinery, by saying we're going to make the immune system do a better job than it's naturally equipped to do so by giving it the help of taking away one of those blocking molecules. So I'm going to stop and turn it over to the other speakers at this point, but what I want to leave you with is the idea that cancer is the result of genetically unstable cells that have acquired abnormalities, don't die off the way they're supposed to, stick around and continue to make more and more mistakes, some of which lead to malignant growth. We need to, the way we stop from doing that, not develop cancers, is by carefully regulating that process. And as we get older, we start to make mistakes, and our cells make these errors, don't get rid of the cells the way we should, and so cancer develops because the weeding out of the genetically undesirable cells isn't taking place as effectively as it should. And the best route forward to dealing with cancer is going to be to understand the fundamental genetics of each of those cells, develop tools that capitalize on the genetic uniqueness of each of the malignant cell, and develop tools that allow us to selectively attack it. Thanks for your attention. I'm going to be around to answer questions as the other speakers have finished up.